It is always a pleasure coming back to UCLA. As Jerry said, I've I got turned on to immunology, started my first research lab here 46 years ago today, or this year. So it's really a pleasure coming. And by the way, I'm not the only one from that class. Dan Portnoy, who's on National Academy, was also one of my good friends in bacteriology back in those days. So anyway, I do love UCLA, and it's a pleasure to be back here. Now, one of the 46 years is a long time, but one of the benefits of that I can enjoy now is that I've been doing this for a long enough time that I'm not going to just talk about an individual research project. I'm really going to try and paint a story for you that has developed. I've been fortunate to be one of the largest non-human primate labs in the country for the past 20 years. And over the years, we've just developed basically a data-based storytelling where we're gonna understand how CD8 T cells work, I think, from the view that I'm going to give. And so that's my goal today. Obviously, I work on SIV infection to develop AIDS vaccines or HIV cure therapies. But to me, this is really the benefit of this work is actually the insight into we would get into basic immunology. All right, so this is what the immune response sees at the peak of viral replication in an SIV infection in Rhesus macaques. And as you can see by all those brown spots in there, those are virally infected cells. And given the fact that this is a highly immune evasive virus that replicates, has massive replication, has high mutation and functional plasticity, you can see where the problem lies. By the immune system is starting at this point, it's already behind the 8 ball. And so it's really it's really a daunting task for the immune system, and particularly the CD8 T cell system, to stop this virus. These responses are usually overwhelmed, and if they're effective, they usually precipitate a mutational escape, which leaves the virus hardly touched. So this has been the problem with both HIV and SIV counterpart in monkeys for the last 40 years. And the primary problem is the one I just told you. It's really the fact that the immune responses come too little, too late. And you don't know when you're going to get exposed to HIV or SIV, and so the system has to start at that first infection, and by the time it develops an effector response, T cell effector response, which is a lot quicker than an antibody response, there's already peak viral replication. The virus is malleable, mutational escape, and this is even before you start thinking about the fact that the virus kills CD4 T cells, which messes up the immune response and can go latent and hide invisibly in tissues, in tissues without the immune response even able to see it. So we haven't even gotten to that problem yet, and we still have a major problem. However, what has been fortunate and has allowed us some opportunity to learn is that even though the immune system does not manage to completely or stringently control this virus or even come close to clearing it in the vast majority of cases, even in the average infection, the CD8 T cell response has a major effect. And do I have a? Oh, good. So if you look here on this figure on the right, this is a standard infection in which half of the monkeys have been depleted of CD8 positive cells. This includes both T cells and NK cells in this study. And you can see that the viral replication rates, which normally come down to a plateau phase, a rate of viral replication that leads to AIDS in about 1.5 years to 2 years, these guys all have 2.5 logs higher, and they all succumb to AIDS in the first 3 or 4, maybe 5 months of infection. So clearly, even in the average case, there's a beneficial effect of the CD8 T cell response. And if you look at, in certain circumstances, most notably animals that have protective MHC alleles, you could see even better than that. You could see animals that become what we call elite controllers, viremia controllers or elite controllers. And these animals can go for years without getting AIDS. And of course, they're also subject to immune escape, in which case they go back to the drawing board of these guys. So there is clearly an effect of the immune response that gives us a sort of an end to be able to study what goes right and what goes wrong. You could also, over the years, there's been many kinds of vaccines that have been developed, standard vaccines, prime boost vaccines, and they also can have a couple of logs. In general, they, you can show some diminished viral replication. Sometimes it's transient because of immune escape, but clearly there's an effect here that you could have by having a prepared CD8 T cell response. And when you look at it closely, and this is what we do over the years, these elite controllers are actually pretty interesting because if you look at where the virus is in these elite controllers, the little residual virus is actually sitting in the B cell follicle, in contrast to a progressor where the infected cells are both inside and outside the follicles. And this linear staining here is dendritic reticulum cell virus. It's not infected cells, but there are infected cells both in and out of the follicle here. And what happens in elite control is that the infection gets driven in completely to the follicles, and this is because CD8 T cells are excluded from B cell follicles for functional reasons. And so what's interesting is that the paracortex, or the non-follicular areas of an elite control, are basically cleared of virus, which is pretty good. And in fact, you can even make it a little better, and I'm showing you this because it comes back to, later in the talk, is that if you actually deplete B cells and get rid of follicles, you can at least transiently even increase the control or decrease the viral replication in animals that have elite control to start with. So the system can work pretty well, and if all the right things come together. And in what I call the cure world, which is basically trying to prevent rebound after infected animals or people are put on antiretroviral therapy, it's even easier to see an effect of CD8 T cells. And this here shows the difference between a primary infection with SIV in the black versus the same virus, the same dose, the same everything, but these animals were put on ART at day 12 and then allowed to go for a year before they were released. And you could see that these guys here, before they were released, you could actually see they're not in black, but in reddish color. That's actually the same guys as these. So they would have had the same primary infection courses here. But once you put them on ART and release them, you have a 2-log difference in viral replication. And we've shown using CD8 beta depletion, which only gets rid of CD8 T cells, that 2-log difference is basically mediated by CD8 T cells. So it's not a home run, but it's a solid single. The CD8 T cells are participating in control of viral replication. And of course, we'd like to understand and make this better. Okay, so what I'm going to tell you today are 2 stories that have, one of them more mature, one of them more mature means it's 20 years, and the more recent one is probably 10 years. And I'm going to use the Rhesus Macaque model. And the model I'm using here is a model of Indian origin Rhesus macaques with SIV MAC239, which is sort of like the death star of SIVs. It usually causes AIDS between 1.5 years and 2 years in the average progressor. So it's probably about 8-fold faster than the average HIV. And the viral loads tend to be, median viral loads are around 1 million or so, depending on how you give it. And so it's quite a high bar. It's highly replicative, it's highly fit, it's highly pathogenic, it's chronic progressive, immune evasive, and neutralization resistant. So that's the high bar that we're working with. And the 2 stories I'm going to tell you is one about, as Jerry mentioned, about our work with cytomegalovirus vectors, which is an entirely new immunology, and then more trying to harness conventional T cell immunology for cure therapy, or post-rebound remission, although apparently that word is not supposed to be used anymore. You're not supposed to use remission. So anyway, so in thinking about a prophylactic vaccine, we have to think about what happens during primary infection. And I have 4 steps here. There's mucosal translocation, their initial cells get infected, there's local replication, there's early spread, and broadcast of the infection, and then finally there's generalized systemic replication. As I've already mentioned to you, just on theoretical grounds and on the basis of observations, there's a line of no return here, where if the immune response comes in here, you're basically sunk, and you're sunk because it's up here in terms of our replication, and the immune escape capabilities are huge. There's also the fact that it takes a while, and I'll show you some of this data later, it takes a while to establish a long-term reservoir, but once you do, you're going to have latently infected cells that are able to reignite the infection at any time. So you want to hopefully get your immune response going before that. So the idea is that you have to consider, well, what kind of immune responses can we get that'll intercept this infection at the right time before all this bad stuff happens. Oops. And so, well, just thinking about theoretically, obviously, if we have pre-established antibodies that are antiviral, neutralizing, and so forth, that would be great. This is a whole other area. Everybody else in the HIV vaccine world works on this, but me, and that's fine. But they haven't exactly figured out how to do it yet, and we're not going to talk about it later. But there are a number of barriers to making those antibodies that are going to work. If you do here, the central memory response, which requires an anamnestic response in order to get effectors, that's the one that's too late. That's what I've showed you thus far. And so the one possibility for actually intercepting this before this red line is what I call pre-positioned effector differentiated responses. These can be what I call effector memory responses, although that includes things that are both in the blood as well as in tissues, which could have been resident memory responses. So it's basically pre-positioned T-cell effectors. And so we made the conclusion a number of years ago that we were going to shoot for this pre-positioned effector differentiated T-cell responses. And I had got the idea back in the late 1990s when I was still in Texas by studying CMV that this is the one infection that naturally does this. If you look at the very high frequency cells that are CMV, that react to CMV, they're distributed in all the right places across all the tissues in fairly high levels, and they have this effect CD28 negative, CCR7 negative effector memory phenotype, particularly CD8, which is dominant. And so these are all granzyme positive. They're all able to manifest a very potent immediate effector response. And of course, that's how they keep CMV, which is a virus that lasts forever, in check. So the question is, could we harness this biology to make an HIV vaccine using the SIV model? And so the original concept that guided the development of CMV vectors was that we knew we had this high response that was distributed across tissues. We knew it was effector memory differentiated, and so we thought that this effector memory response could intercept the infection here and basically do a whack-a-virus thing, and maybe stop the infection before all the bad things I mentioned happened, and that was the hypothesis. Sounds like it makes a lot of sense. And so we set out to answer the questions, starting in the mid 2005, 2006, 2007, started making these vectors and asked whether a Rhesus CMV best vector. Remember, CMV are species specific. So in Rhesus, we use Rhesus CMV, and humans use HCMV. But whether this vaccine would be able to do better against a mucosal challenge with our Death Star SIV MAC239. And lo and behold, it did. Okay, it was almost magical, in fact. And this is just one example. We've done hundreds of monkeys now, and you'll see how this evolves in a few minutes. But this is just one example of such an experiment where animals that were vaccinated were challenged with repeated limiting dose SIV MAC239 down, challenged until they were demonstrably infected. Remember, T cells don't prevent infection; they can only stop it once it happens. So what we would do is we'd monitor viremia, we would monitor for viral T cell responses against like SIV VIF, which is not in the vaccine. You could use anything that's not in the vaccine for this. And we asked the question, once the animals got infected, what happened? And it turns out about almost 60% of the animals had this very unique phenotype where they sometimes had a blip of virus, sometimes not. They always developed responses to SIV, VIF, or whatever that wasn't in the vaccine. So they clearly had the immune system was seen wild type SIV or the challenge virus. And in fact, if you look in tissues, and this is bone marrow because it's easy to get to, you'll find cell-associated virus that that isn't now, at least initially, isn't all that less than you see in the black ones, which are progressive infection. And if you take cells from these guys right here, and there's blood cells or lymph node cells or what have you, and you put them in another animal, we call Doppler transfer experiments, you get fully pathogenic SIV out. So these animals are undeniably infected with SIV, but the SIV is arrested in the tissues. It's what we call replication arrest. And the interesting thing is that if you look over time, the VIF responses go up, they come down, they go away. The ability to detect virus in tissues goes away until, I'll show you, until it's completely gone. And in fact, you can no longer adoptively transfer infection, after a year or 2 years after they were initially infected. So it looks like they cure themselves. And in fact, if you look at, here's a necropsy study that was done, whatever, shortly after, I can't see it, PID 22, PID 39. It's easy to find virus. You find it in the portals of entries. You find it in draining lymph nodes, the portals of entries, and you find it early hematogenous sites, liver, spleen, and bone marrow. I already showed you about bone marrow. But if you wait until, this is PID 532, PID 483, there's no, you often find animals with no detectable virus. So this works. Most of the animals we use are CMV positive because animals are, these monkeys in colony raised are CMV positive shortly after birth. But we especially raise CMV negative animals, we see protection in both. So pre-existing immunity doesn't matter. Pre-existing non-immunity doesn't matter, it works both ways. You could also take these animals after they've completely cleared the infection. So they go from what I showed you before, which is adoptive transfer negative, and then you re-challenge them, and then you can get them to recontrol the virus. And they turn adoptive transfer positive, but they have replication arrest. And we've done this as long as 11 years after the initial vaccination. Okay, so this is a very different kind of protection. Here's the protective allele, conventional CD8 T cell response to epitopes that are restricted, that have limited malleability, so that the T cell responses that target them tend to work better. This is what it looks like. Obviously, they intercept too late to stop peak infection, but then they can control the virus until it escapes. This is a very different situation. These guys have a blip or no blip, but they get infected and then they're completely replication arrested. There's no escape. These guys just don't get protected, but they have no immune escape. They're not escaping from the virus. The sequence hasn't changed. It's way too early for that. It's just like it's an all or nothing kind of protection. So what's the mechanism? Well, first of all, we can rule out antibodies, and the easiest way to rule them out is the fact that these vectors don't make antibodies to the inserts. Okay, or certainly most of the animals don't make antibodies. Sometimes they have a little bit, but basically you could leave out envelope. You could do this just with GAG. We tend to use GAG, 5' Paul, and RevNev-TAT as our sort of go-to inserts. You don't need envelopes. Basically, there's antibodies are sort of ruled out by those two observations. And in contrast, if you do CD8 beta depletion and maintain the CD8s low during the challenge time, you actually aggregate protection. So it definitely implicates CD8 T cell responses as being the mediator, or at least as required for the efficacy. So is it just robust TDM? Well, we've tested that in a number of ways, and I'm going to show you one here. We've actually, with Tom Henke, his ChAdOx MVA vaccine is pretty potent, and in fact yields magnitudes that are not particularly different from the CMV vectors. It's shown in blue. This is the red ones, and the ChAdOx ones are almost completely effector memory. Yet there's no replication arrest protection in these guys, where there is in these guys. And in fact, these really effector memory vaccines, and we've done this with the prime boost of David Mousefus, we've done this with his ChAdOx, they actually don't work very well at all. They don't even have the usual log or two of protection. So there's something very special about these CMV responses that is not replicated, that is not just based on the differentiation status of the cells. Okay, so this is where it gets a little bit weird. We said, okay, well, let's figure out what this is. The first thing you do is, well, what can we measure? Well, let's see what the epitopes look like. And so the first thing we did is we looked for all the usual suspects, which are the immunodominant MHC1A restricted epitopes that are classically elicited by vaccines or by SIV itself. And there's a whole list of them here with different, that are for different MAMO-A01, MAMO-A02. And basically, none of these epitopes were seen in these vector. You see responses to the whole insert, but not to these epitopes. And in fact, if you do over here on this side, this is a prime boost experiment. When you start with MVA gag, and you come back with Rhesus CMV, you don't see any boosting of these pre-existent ones or electroporated DNA, no boosting. You see new epitopes being generated. So clearly the epitopes that are targeted with CMV vectors are completely different. And to make a long story short, we went in and we basically did 15 more consecutive peptides. We 11 amino acids overlap. We just looked at them all across the gag protein, and whoa, look at all these epitopes. Each box represents a response, but when we blocked them, they were blocked either by class 2 antibodies or by VL9, which is the MHCE specific peptide, and none of them were MHC1A restricted. And this was confirmed with transductants, showing that these particular responses were restricted by either class 2 or by MHCE. And I'll tell you what MHCE is in a minute. The other interesting thing about these viruses is that they're supertopes, which are epitopes that are present in both class 2 and MHCE, which are present in 100% of animals. And this is pretty weird, but this is unequivocal what was happening. And we didn't know initially where this response came from or whether it had anything to do with protection. So let's start with the first question. It turns out that this response came from an extremely lucky mutation that occurred in the Rhesus CMV that we initially used for this vector. This is 68-1 Rhesus CMV. This is a virus that had been multiply passaged prior to making it back and make it so we can make it into vectors. Like most CMVs, that are grown in fibroblasts, it had a mutation to remove what's called the pentameric complex, which the only thing you need to know about it from this perspective is that slows down growth on fibroblasts, and so the virus loses it. And so, but it had a complex inversion deletion, which not only took out the pentameric complex, but took out another set of genes on the other side here. It turns out that this was a surgical strike from mother luck, because what happened is that we figured out, yes, if you corrected this, if you went back to wild type, you went back to MHC1A restricted T cells. So it was perfectly normal T cells, none of this funny stuff. And we went in, and of course, we've basically gone through the genome now to determine the genetic origin of all this and looked at all these genes individually. And to make a long story short, that the Rhesus CMV and HCMV encodes, well, Rhesus CMV it's 8 and HCMV it's 4, encodes inhibitors that proteins they're both two components of the pentameric complex, as well as this UL 146, 147 orthologs, which are chemokines. What you need to know about them is only that they inhibit this unconventional response. The virus doesn't like this unconventional response. It has evolved in the Rhesus 8 different genes to stop it. Okay, and if you put them back, any one of these genes back, at least some of these genes here, that you get completely back to MHC1A. If you put back these genes here, you get rid of the E response and still have a mixture of class 2 and class 1 response. We've also discovered that there are genes that are required for these responses. So there's a gene called UL40, Rhesus 67 is the Rhesus word for it. That is a gene that is involved in upregulating MHCE on the surface of the cell. If you get rid of that, you lose the MHCE responses and you only have class 2 responses. So we've now gone through and basically gone through the genome, and we've identified almost all of the genes, and I'll show you how we know that in a minute, that are required for this type of response. And then the other thing we've learned is that viral, and here's the list of the genes. There's also one we found in HCMV that doesn't have an ortholog in Rhesus CMV. So HCMV has the orthologs and an additional inhibitor. So the viruses really don't like these unconventional responses. Anyway, those are the genes we've identified. But even more interesting is we've also identified that these responses depend on CMV diverse viral tropism by using microRNA restriction, which basically makes the virus so it's susceptible to intrinsic cell-specific microRNA types, so it can no longer productively infect certain cell types. You actually modify these responses. So if you block endothelial cell infection, you block the class 2 responses and get only MHCE responses. If you block myeloid cell restriction with MIR142, you get just the opposite. You get class 2 responses only. And if you block both, it reverts. These are direct presentation. If you block both, it reverts to class 1 again. So yeah, and this virus is identical except for these tropism restrictions. And and to make and basically this gives us an opportunity to say, well, what do these different responses do? What they all look the same in terms of magnitude, breadth, and effector memory differentiation. What's protective? And it turns out that if you take all these vectors that are programmed to make non-MHCE responses, none of them work. So only the original vector and the MHCE only vectors are capable of replication arrest. So this clearly indicates that this MHCE restricted T cell response is the secret sauce that makes this work. Okay, so what is MHCE? MHCE is one of the most conserved MHC1 loci in primates. It's relatively non-polymorphic. In humans, there's basically 2 polymorphisms. In Rhesus, there's a few more, but they're not functionally polymorphic. The changes are all outside the peptide binding region. Their job is to prevent a 9-mer VL9 from the leader sequence of MHC1A alleles on the surface to NK cells, primarily CD94 NKG2A, which basically tells the cell, a normal cell, that this normal cell is making class 1 and leave it alone. It's an inhibitory receptor. When a virus comes in or a neoplasm comes in and wipes out MHC expression, then this no longer happens, and the NK cells are primed to kill the cell. Now, viruses like to evade NK cells, and one of the interesting things about this is they tend to upregulate MHCE when they downregulate MHC1. This is for showing for SIV. I mean, with CMV, it's very explicit. They upregulate MHCE hugely, which is why we get these responses in order to evade NK cells. So basically, what we're doing is MHCE responses sort of take advantage of immune evasion for NK cells, and said, okay, well, you're going to do this evade NK cells. We're going to call your bluff and make CD8 T cells recognize those epitopes. Some interesting facts about the MHCE T cell presentation is that the peptides, the exogenous peptides that bind really weakly. They still work. Depending on which assay you use, different ones bind. The red lines here show versus VL9 in yellow. Here's VL9 here. They're relatively weak. Some of them you can't even measure by this assay, but you can measure by this assay. So it's really weak peptide binding. The interesting thing about it, if you sort the cells, if you take cells and you put them in vitro and you give them infected cells to respond to, and you sort the activated cells by whatever criteria you want to do, it doesn't matter, you get the same answer. You find that the TCRs that are involved in this response are relatively few. In this particular example, there's 4 major ones and a bunch of minor ones. And if you clone those TCRs and look at recognition, you could show that they in fact do recognize SIV infected cells. But then when you look at their characteristics, you find that they're very cross-reactive. They tend to recognize at least one supertope and often many other epitopes. And some, so here's sorting cells in GAG120 responding, and this is one TCR that does all of this binding. So they're very cross-reactive. And here's another one with a little more complex patterns. You can see that TCRs are shared across these different epitopes. The other thing about them is that they're relatively weak compared to a classical MHC, which really TCR transfectant. These things are much weaker. But they still can fire with response to their peptide. So I told you there's a lot of breadth in these responses, but the breadth doesn't matter because we've got limited breadth and see the exact same. If you limit the inserts, you see the exact same protection. And again, there's the size of the responses doesn't predict protection. So the MHCE responses tend to be the same in protected and non-protected animals. And I just want to point out here one other thing. This is a recent compendium of a lot of different studies we did. The overall efficacy is 59%, but it's the same whether you have MHCE alone or whether you have the mixed class 2 or MHCE responses. So it appears that MHCE is restricted T cells are required for efficacy, but in animals that have those responses, they don't determine efficacy. Something else must be necessary. So to make a lot of work, sort of long story short, we went to work with Michael Gale's group, who does transcriptomics, and this is whole blood transcriptomics change from baseline, from pre-vaccine to post-vaccine, And you can see that protected animals have a different response pattern than non-protected animals in 2 different cohorts here. And one of the things that we saw was that IL-15, the computer said, was an upstream regulator of many of these genes, and in fact, even IL-15 itself almost reached significance in terms of differential expression of protected versus non-protected animals. And so I said, I wonder if this has anything to do with IL-15. I would point out that the response, which is an innate response to vaccination, is maintained until challenged. There's all these different time points here. And in unprotected animals, you might initially do it, but it's lost over time. So the difference seems to be in keeping this sustained response going in terms of being protected. And then we asked, well, what about IL-15? And we did IL-15 on monkeys that we just gave IL-15 to, and it turns out that it's the same genes that we see in the protective signature. So a lot of these genes are IL-15 regulated, and this was validated in a separate cohort. So IL-15 must be upregulated and sustained. Upregulated and sustained IL-15 signaling is correlated with protection. It's not the only thing that's correlated, but it seems to be a major component. And interestingly, if you look back at baseline, it turns out you have exactly the opposite score. The animals that are protected in red are actually lower IL-15 in terms of upregulated expression of upregulated genes, or higher for downregulated genes. So basically, the animals that are ultimately protected ultimately have the high IL-15 response, or IL-15 quiescent start, which is important. So you have to be IL-15 quiescent so that you can have a big IL-15 response so you can get protected. Okay. And just to show you that we sort of got a handle on what's required for this, Jonah Sasha, who was also at HSU, took this to an entirely different monkey species, cynomolgus macaques. And it turns out that the vectors, you have to use a cynomolgus vector. You can't use a rhesus vector. It doesn't work. But if you use a cynomolgus vector that's designed the same way, gets rid of the inhibitory genes, you get the same kind of protection, replication and rest protection, and it correlates again with IL-15 signaling. So this, we've translated it to another monkey system. If only monkeys had health insurance, we'd be in good shape. So, to summarize this part of the talk, which I'm doing here, okay, replication arrest efficacy therefore appears to require the following: MHC-restricted T cells. These are supertopes and other epitopes that are very loosely bound, relatively few, low avidity, highly cross-reactive TCRs. We think the high magnitude and intercept tissues and the effective differentiation is important, but it's just not the only thing that's important. And we also need this sustained innate immune response. Okay, so I'm going to spend just a minute sort of speculating on why this might be, how we put all this together. So, we need IL-15 signaling, we have these weak T cells, and yet they seem to work different than classical T cells, which are much stronger. So one possibility, again, these are speculative, or we're trying to get data for it, is that it's simply the T cells that are MHC restricted recognize early infection better. And they recognize early infection better because MHC is upregulated, whereas MHC-1A is downregulated, and MHC-2 is not expressed on resting cells. So more infected cells are going to be, say, waving MHC-E than waving into the other MHC molecules. So it could be that ability to see the entire spectrum of infected cells is what makes the difference. That's one possibility. Another possibility is that that's not, it could be in addition to or separate from, is that when you actually look at or imagine what happens with conventional CD8 T cells that recognize class 1, I mean, they cause a lot of mayhem. They kill a lot of cells, but they release a lot of cytokines. They bring in other cells that are target cells for SIV, and basically they can't kill the infection fast enough to stop it, and the virus sort of loves to live in this tumult. It loves to live in this immune activated environment. On the other hand, the MHCE ones, because they're very, their thresholds are high for activation, and because they don't tend to be all that cytolytic, they probably just make things like beta chemokine, which has a very low threshold for production, and they're sort of stealth. They create a field effect where the virus can't spread without causing a lot of mayhem. And the reason you need IL-15 is that if you don't have IL-15, then you don't even get that response, and therefore the animals don't, the infection just goes on. So these are just ideas, but it's interesting. May the meek inherit the earth. The weaker T cells seem to be more effective in this case. Okay. I just want to point out that I have a 2023 review article that goes into all this if you're interested in following it up more. Okay. Will this work in humans? I don't know. We've made a rhesus ortholog of a human vector that's currently being tested by Vir. It works in monkeys. We will see whether Vir can do this without can make this work. They, founders of companies get kicked out, so I don't have much say over that or control. So we'll hear from them as this goes forward. So I'm going to switch gears here. Here are the gears I'm switching. I'm gonna in the last few minutes, we're gonna go from prevention to therapy. And again, as I showed you, usually you get in after you put animals on ART, release them from ART, infection and recrudesces. And even though it's a little bit less than it was before, it's still not enough to prevent progression, except for sometimes it's controlled, these so-called post-ART controllers. And I just want to remind you that post-ART viral rebound is a very different beast from primary infection, as opposed to primary infection, which is a relatively limited number of 1, 2, 3 trajectories of infection. This infection comes from anywhere. The infection is located in all tissues, and as I'll show you, there's lots of trajectories of which this infection can come from, and recrudesce infection. So the first, and here, this is just showing how reservoir works in monkeys. These are animals that are put on ART at various time points, day 3, 4, 5, 6, 7, 9, and 12. These are the relative area under the curves in terms of how much infection they have. And then here is barcoded. These are viruses that in which we have about 400 different species that we can measure because of barcodes. And so we could actually use the barcode to determine reactivation rates. And it turns out that early on, there's no reactivation. So early ART, in this particular case, you do not establish a long-term reservoir. This is after a year of ART. You could find virus for 6 months. You adoptively transfer it, but then it seems to go away because it's not long-lived, except for this one animal. And then there's an exponential increase as you go to about day 9. And then once you're at day 9, you basically have a full reservoir. So the reservoir, what we call the rebound competent reservoir, is actually saturable. And it saturates right about here. So anything, and if you look at animals that are in this category here, there's no correlation of anything you measure about the reservoir and time to rebound. There's differences for sure, but that's determined by other things like maybe innate immunity or something, not by the level of the reservoir. So that's what we're dealing with, and it allows us to do a model with full reservoir, allows us to do a model with a limited reservoir, and that's why that's how we go. So the first question is, would the Rhesus CV vectors work as a therapeutic vaccine? And we tested that a number of times. We can certainly show that here's animals that are on ART. We can certainly show that they get infection, they get responses to their gag responses are increased, and we can see these supertope responses in all the animals, so that the vector seems to work, makes it unconventional responses, but in terms of immunogenicity, but not in terms of protection. So these are animals that were put on ART at different times. Some of them didn't come back because the reservoir wasn't established, but there was no difference between either the time to rebound or the what happened after rebound in Rhesus CV vaccinated animals. And this has been repeated in the full reservoir study as well. So it doesn't seem to work. We don't know why, if this is just because you can't get IL-15 programming to work because you have an infection and you're not quiescent for IL-15. We don't know if it's just too many foci, and that that CMV vectors can handle 2 or 3, but they're not going to be able to handle a continuous assault of virus infection. But for whatever reason, it didn't work, making us go back to the drawing board. One advantage we have with this scenario, where we don't have a primary infection, is that we control when this occurs. And monkeys or in people, when you release ART is when you're going to have this. So you can actually plan for this and do immunotherapies that will hopefully do better than the natural response. And so I already told you that CD8 T cells already have been shown to have a response, have an effect on plateau phase viral loads with no effect to time to rebound, at least the natural response. There's no effect to time to rebound. If you look at MHC protective alleles, animals that have protective alleles and animals that don't have protective alleles have no difference in time to rebound, but there's a difference. The protective alleles lead to a lower plateau phase viral load. So again, T cells seem to come in and after the fact and do something, but not enough to stop the rebound altogether. But so what we wanted, even in those situations, we know that the T cell response is still following viral replication. It's following it a little bit more closely than it does in primary infection, where it has to start from the naive T cell system, because there's memory T cells here, but it's still following. And so the question is, can we move this orange thing back? And can we have the CD8 T cell responses already sort of stimulated prior to art release in order to make this work a little better and get post-treatment control. Okay, so I'm gonna, the other thing that shows this is probably important is that if you look at, again, remember, T cells don't determine time to rebound, but time to rebound actually correlates with, post-art control. Again, supporting the concept that if you can get the T cell response ahead of the, in this case, by having rebound for whatever reason, maybe innate response is being delayed, if you can get the T cell response time, it'll work better and lead to, whether or not you have MHCLEs, it'll work better and lead to better post-art control. Okay, so start with some experiments. PD-1, checkpoint inhibition. We've shown that PD-1 can actually increase control in animals that have elite control. So clearly, you can increase the effector efficiency of RMCD T cells. Can this mediate post-art control? We did two experiment, one where the PD-1 was started 8 weeks prior to art release, and one was started right at art release. No difference, just like we saw before, no difference in time to rebound. So this didn't help the intercept. But it did, as long as you were given the drugs, it helped the control. Of course, once you stop the anti-PD-1, you just go back right up, which is about right here. You just go back right up and you're back where you started. So this is basically more, but still too little, too late. Another possibility is we talked about B cell follicles. I showed you that if we got rid of B cell follicles, would that help? So we did this study with anti-CD20, which disrupts T cell follicles. There's two types of monkeys here. One with B08, which is a protective allele, the others without a protective allele. And in fact, we showed that if you do see follicle depletion, you actually have a slight effect on viral rebound. It's a little bit delayed and it's a little bit controlled early, a little bit later, but obviously once the CD20 is stopped, it goes back to where it was. And that only works when there's effective responses. There wasn't a statistically significant effect when there's no effective response. So this therapy showed a little, but not a lot. And in fact, we tried to improve on that by giving NA20, which is a combination anti-CD20 and IL-15. So we figured, oh, this is really good. We'll give it, we'll stimulate the T cells with IL-15. It also stimulates NK cells, so they get rid of the B cells even better. And basically, all the follicles are gone with this therapy. And so we thought we'd be really smart. But what we forgot about is that CD4 T cells are hugely stimulated by IL-15. And in fact, this abrogated the protective effect. And in fact, we had an increase, at least transient, higher viral loads in the ones that were treated at the time of art release. So, you need to, you don't want to go too far in terms of this, immune stimulation. Okay, so how about just the T cell vaccine? Okay, well, regular T cell vaccine. Well, that makes sense. We like T cells. So we have an mRNA vaccine that we've developed that in this particular case expresses GAG. It turns out if you give it 1, 2, 3, 4, and then one time before art release, you get really big responses. This is in lung wash, which is really nice because it's an effector site that you can reproducibly access, and you can see that you're making tons of effector cells with this vaccine compared to the animals that were left on art alone. And this is on art and off art. You can see that this response, is much higher in the early times off art because you basically started it before the animals were released, where this is still following the infection. So what happened? Oh, well, one more thing. This is in all tissues. These are actually tetramer responses in various tissues Again, the blue ones are the animals that had tetramers. You can see that the responses are much higher in almost all these tissues. So that looked pretty good at the time. This is at the time of ART release. And sure enough, we saw an interesting effect. First of all, this is the first time we've seen a delay in time to rebound. So T cells' natural responses don't delay rebound, but if you really boost the responses at the time of ART release, you can delay time to rebound. And in fact, but we didn't prevent rebound, unfortunately. And we had an early significant effect on viral replication rates. But as time is going on, and we're basically out here right now, they're starting to merge. So basically, this is not a home run, but we are showing that additional viral control mediated by a therapeutic vaccine was not sufficient to, like I said, to stop rebound. But it does support the concept of a more potent immune intercept, more potent immune intercept can have positive effects in terms of delaying rebound and controlling post-rebound viremia. Okay, I have a couple minutes left, and I want to show you something really interesting and really recent data. What I've shown you basically now is up until now in terms of was basically an empirical testing of all sorts of things that worked in other systems, cancer, whatever, and we just tried them to see if they work. I like to do big projects and basic projects, and so ask the question. Maybe we should try to understand how rebound works first, so we can know where it is that we need to target, and what the environments are that rebound occurs. So we wanted to ask whether we could figure out where the virus comes from, what tissues does it come from, how does it spread after ART is released. We want to know what are the processes by which initial tissue rebound is locally and normally systemically amplified or restricted. And basically, what is the role of local neighborhoods? So we want to find the rebounding virus, and we want to understand what the local immune response is to those rebounding viruses. Why some animals take 4 weeks to rebound, and other animals which end up doing better than animals who just take 7 days, and so forth. Basically, this territory here in pink, which is what's happening in tissues after ART is released, but before overt viremia, there be dragons here. Nobody has really looked systematically at this and basically said, how does these changes occur? How do you go from on ART to off ART initial rebound? What is the first rebound? And to do this, we planned a serial necropsy study. These are very expensive, but very time consuming. In this particular study, we had 24 RM, barcoded SIV MAC239M, a full reservoir, ART starting at day 9, and we did necropsies on ART for the control. We have to know what the baseline is, and then we did 18 animals that were necropsied either at day 5 or day 7, because day 7 is the median date for time to rebound in untreated animals. And we actually hit pretty spot on. Here's the blood viremia of the animals that we took to necropsy. Most of them were obviously most of the ones on ART were aviremic, but most of the other ones were aviremic, and there were a few that had low viremia and one that had overt rebound. And basically what we did is we collected 400 tissues from these animals, and we divided each tissue into 3 different parts: tissue chunks in which we did viral RNA DNA quantification, barcode sequencings, and bulk transcriptomics. Again, the transcriptomics is trying to understand what the immune response is. We took another one that was formalin fixed for histology. Jake Estes is going to do spatial transcriptomics, RNA scope. Again, but all of these different had both viral detection and transcriptomic analysis. And finally, these are single cells, which we did single cell RNA-seq, as well as cell-associated viral load. And this study is just in its infancy, but it's really been interesting so far. So real quickly, what are we looking for? How do we know that there's rebound? What we're looking for is local tissue spread. On ART, you can get a viral expression, as shown here. You can get RNA being produced, but what we're looking for is that those first cells that are not the original latent cells that are hosting infection. And what we know is that the RNA DNA ratio on cells that are actually going through the full range of infection, making new virions, is much higher than it is when they're on ART. So one of the things we can look at, and this is just peak day, this is day 12, this is off ART. You can see that these cells are packed with RNA. So one of the things we're looking at is the RNA DNA ratio. We do that by Cavill. We can look for RNA content per cell, like for here, RNA scope, and we can also look at the density and distribution of RNA cells for RNA scope. And so those are the criteria we look for. Just to start with doing RNA scope, as you can see here, this is an example of a lymph node off art. And if you do serial sections, basically you can see where the reds are the infected cells, and then you can see that we're finding clusters of cells in this off art animal. And we don't see those clusters like that on art. And this is quantified here. You can see that the on art, this is the distance between infected cells, and basically they're infinitely distant and on art, whereas on all these off art aviremics and viremics and so forth, you see clusters of cells. So the other thing you see is an increase in RNA content per cell, even in the aviremics off art. But the more interesting data actually concerns barcodes. And again, I know that Jerry does this in his lab, but you basically create false, basically isogenic heterogeneity, so you could follow clonal lineages of viruses by sequence. So these viruses are identical except for in terms of protein expression, but they have different barcodes that allow them sequencing to distinguish different clonotypes. And there's typically 200 to 400 some odd in the animals that we using our system. And you could actually follow, like for example, here's rebound showing 3 clonotypes coming back from this animal that was taken off art after starting early, but having full rebound. So you could actually use that to track the clonal contribution rebound. But what it hasn't been used for, and what we're using it for now, is really tissues. And this is where it really gets interesting. So these are anatograms here. We started with just 50 samples. We haven't done all 400 yet. But as we look in these 50 samples, and this is an animal that had a PVL 65, so it's low viremic rebound, off art at day 12. You look in, I forget what tissue this was, this is somewhere in the gut, the colon, and you can see that these blue, this is DNA, that red is RNA. You can see most of these DNA species have no RNA at all, and some of them have a little bit of RNA, but then 2 of them have very high RNA to DNA ratios. And it turns out that if you look at those 2 barcodes and just look at them individually, you can see that this one is sitting in that tissue has a 1,000 to 1 RNA DNA ratio. There's no question that's spreading. And it also this one contributes 2 of those CPM in the blood. And this one is now in 4 different spots, 2 very high. And it's the one that's contributing 50 copies per mil in plasma. And you can see that the rest of these tissues are basically negative for this virus. So these clonotypes. So basically, we're actually capturing early rebound. We can go back. And look at animals with even lower viral loads. Here's 28. Here's an ileocecal lymph node. Again, cells with viruses with no RNA, some with basically even RNA to DNA, and then a couple with very high RNA to DNA ratios. You could do the same sort of thing, and you could see that these barcodes are basically in this one tissue. So this one tissue is where the rebound appears to come from. There may be others. We're doing 50 samples at a time, but certainly this seems to be a very paucifocal rebound site. And of course, we can compare the sites that don't have rebound to the sites that have rebound to determine the immunology of rebound, since we have all those samples. If you look at just the random tissue that doesn't have any evidence of higher RNA DNA overall, you can see that basically RNA DNA ratios are quite low, and there's nothing going on in that tissue. So you could find both tissues with and without rebound by this technique. And again, even down to 5 copies per mil, which is basically aviremic, you could find excess RNA to DNA ratios on certain clonotypes. And again, 10 or above is clearly showing spread. 5 to 10 is sort of equivocal, and less than 5 in terms of RNA DNA ratio is nothing's happening. So we're hoping to use this to basically take these monkeys and really identify both the virology and immunobiology of viral rebound with a concept that by doing so we could better target or even the therapies. So just the preliminary conclusions. Again, this is a work in progress. Early viral rebound is not a needle in a haystack event that occurs in some special compartment, although it tends to most often occur in mucosal lymph nodes, mesenteric lymph nodes, and mucosal sites. That's something that's interesting because those sites that are not looked at in humans, and that's where it seems to be happening. But it doesn't just occur in a needle and explode everywhere. It tends to, it's a multi, but generally paucifocal process that occurs in various lymphoid tissues, different individuals. It tends to stay local until relatively late in the process. The mechanisms that restrict the local rebound, that have SIV infected cells, the laxus rebound or promote rebound, these are unclear, but we're hoping that the comprehensive omics analysis plan in the study will reveal the pathways that are operating in these local neighborhoods and potentially give us targets for which we can plan therapeutic interventions that could delay rebound and ultimately collaborate with, for example, a therapeutic vaccine to have better outcomes, more post-ART control. Okay, so a little bit late. Final thoughts. You know, I just want to. This has really been a great system to work on. It's been great fun working on. You know, completely successful immune responses are really hard to dissect. It's actually nice to find a broken one. And the SIV, CD8 T cell response in SIV is a broken one. Not completely broken, but broken. And so from trying to fix the system, we've learned a lot about the immune system. And in our work alone, we've learned about a novel immune response, which is now all the rage. I mean, there's just a paper Nature Immunology about MHCE responses to flu and so forth and so on. We've started a cavalcade of papers looking at MHCE T cells, but this is an entirely novel response, which has implications not only for infectious disease, but for cancer. And we've shown that I think unequivocally shown that immune dynamics is critical. It's not just how good your T cells are, it's really the dynamics by which they intercept infection. And that's, I think that this model is really illustrative. Now, monkeys aren't humans, so we can't model all aspects of human infection. But so far, all the mechanisms that we've revealed have clear applicability to humans, and hopefully will help us develop sophisticated new approaches with broad applicability. And with that, I just want, I had to do a new acknowledgement slide, but the key people are shown in pictures, but in particular, Scott Hansen, who runs the CMV vector group in my laboratory, and Afam McCoy, who runs the cure group. Jake Estes, of course, is my new boss and also does all of the Cyto work. Klaus Fu is my virologist partner for CMV vectors. Of course, I've worked with Jeff Lifson for virology for many years. And I mentioned Michael Gale in terms of transcriptomics. And I guess with that, I'll stop and take any questions.